Hello there, and welcome back to Teach Me in 10. This is, of course, a video series that's brought to you by Technology Networks. In today's episode of Teach Me in 10, we'll be hearing from Jessica Hall, who's a field application scientist at Sphere Fluidics Limited. Today, she'll be exploring the acceleration of single-cell analysis and screening within the drug discovery process, showcasing how the revolutionary potential of picodroplets can transform crucial steps. And before we go any further, I am so excited to say that this episode is kindly brought to you by Sphere Fluidics. Sphere Fluidics is the Cambridge, UK-based microfluidics company with a range of novel cell encapsulation products for single-cell analysis and isolation. To learn more about how their technology platforms accelerate biopharmaceutical discovery and development, cell therapy, and synthetic biology workflows, you can click the links that we've left in the description for you. But without further ado, I'm now pleased to hand over to Jessica.
Hello, so just to introduce the face at the top of your screen, I'm Jessica Hall, a field application scientist from Sphere Fluidics, a microfluidic biotechnology company specialised in single-cell analysis and isolation platforms. And today I'll be teaching you in 10 about the power of picodroplets in single-cell analysis and screening and the drug development lifecycle. So to set the scene, when you buy a drug from the supermarket or when you receive drug treatments or drug therapies, there has been on average a whole 12 years of work and $1 billion spent by pharmaceutical companies to bring that product from concept to market. This is called the drug development lifecycle, and it consists of 5 stages. It starts with discovering key molecules that could be effective in curing or treating the target disease. Potential drug molecules are tested and screened for their efficacy against the disease the drug is intended for, so our target disease. The potential drug molecules are then tested in the lab before being sent for clinical trials in humans. And at the start of this process, there are thousands of potential drug molecules. However, through each of the stages, this is decreased until finally yielding one marketable drug, which is sent for approval by regulatory bodies. And during these early discovery phases, as there are so many molecules to test, this is a significant investment of time and money for companies trying to push products through this process. Pharmaceutical companies want to quickly narrow down from thousands of potential molecules to just a few hundred. Therefore, this industry has seen a multitude of technological advancements in laboratory automation to combat the 2 major drawbacks of discovery: time and cost. And one area of drug development that has taken off recently is the world of biotherapeutics. These come from molecules secreted by cells to create immunotherapies to treat diseases such as cancer. Therefore, what these early stages of drug development entail is something we call single-cell analysis, where we analyse cells separately in order to investigate their secretions. So in single-cell analysis, we isolate single cells from a population of heterogeneous varied cells and study the interaction of their cell secretions with our target disease molecule. So, for instance, if we were developing a therapeutic for a virus, the disease molecule would be part of this virus and we would be testing the individual cells to see if they secrete something that binds to our virus fragment. And if it does interact with the virus, this is a potential drug therapeutic that can be used against it. And the reason we want to isolate these single cells is to ensure that the secretions we're testing are indeed from that specific cell and not another cell. So we want to collect the cells that are effective against our target disease. And often, as these secretions are so small, we can't see if they have been successful at interacting with our disease molecules. Therefore, we rely on a fluorescent readout. So for this, we designed fluorescent assays to detect our secreted drug candidates. These assays consist of components which are added to the single cells to produce a fluorescent light signal when a cell secretion has successfully interacted with the disease molecule. Cell secretions which cause this fluorescent readout are then kept for downstream screening. So for cells that secrete non-compatible antibodies, like this cell in the middle, or don't secrete anything at all, like this one on the right, they do not have the right cell secretions which bind to our disease antigen, and therefore they don't produce a fluorescent signal. These are discarded, whereas the cell on the left produces antibodies that are potential drug candidates against our target disease, causing this fluorescent readout. And these single cells are kept for downstream processing through the drug development lifecycle. So how is this single cell analysis done traditionally? This is done by a process called limiting dilution cloning, and it pretty much does what it says on the tin. We dilute our cell population to limit the concentration, which in turn allows us to isolate single cells in the population into well plates. So for example, here we have two heterogeneous populations of cells. On our left is a very high concentration of cells, and on our right is a low concentration of cells. So these cells on the right have been diluted, and this is our limiting dilution. If we were to load a small volume into each well on a well plate, for the high concentration cell population, we would get something like this. Here there are multiple cells in each well, and this isn't what we want as it doesn't allow us to study each cell individually. So if one of these wells were to start fluorescing, we couldn't be sure which cell was causing this. Whereas with the limiting dilution, at a lower concentration, we get single cells in around half of the wells. And this allows us to study each cell and its secretions individually. So we can perform our single cell analysis by adding in our antigen and seeing which cells have antibodies that bind, and then selecting for the few cells of interest via fluorescent readout. However, often to isolate a whole cell population, let's say about 1 million cells, we would need on average 20,000 plates. And this is incredibly time consuming, and the reagent cost is very high. So how can we speed up this tedious process? Introducing microfluidic droplet technology. So instead of studying these single cells in wells on a plate, we choose to study single cells in tiny picoliter-sized droplets. And just to give you an idea of the size of these picodroplets, here is a rough size representation of what the cells look like in one of these wells on a well plate. And here is what a picodroplet containing a cell would look like in comparison. And these picodroplets are created by pumping surfactant oil perpendicularly to your cell sample. So this encapsulates your cells into picodroplets. These uniformly sized droplets form confined microenvironments in which to study single cells. And not only are these droplets a fraction of the size of the well plates, massively reducing reagent cost, they can be generated extremely quickly, so about 2 million in about an hour. And this is no element of manual labour. So this allows for extremely high throughput automated processing. They also have the added benefit of being able to bring all of the assay components in close proximity for increased assay sensitivity and faster fluorescent readout. This process is gentle to cells and allows for novel assays never before possible. So how do we use these tiny droplets to conduct single cell analysis? So Sphere Fluidics collaborated with industry specialists to produce our flagship instrument, the Cyto-Mine, with the mission to find, analyze, and isolate your most valuable cells with ease and speed using microfluidic picodroplet technology. So, starting with a heterogeneous cell population, this is prepared and loaded onto the Cyto-Mine where it undergoes single cell analysis within picodroplets. And what you're left with at the end is plates containing cells of interest in every single well. So these cells will all produce antibodies that bind to our target disease antigen. And this happens in 4 main steps. The sample is first encapsulated into picodroplets, and you can generate up to 2 million of these, allowing you to analyze between 100,000 and 40 million cells. The droplets are then incubated for a user-controlled length of time, which allows the concentration of cell secretions to be built up. The droplets are then sorted via fluorescence readout. Our cells secreting antibodies, which bind to our antigen, will turn the picodroplet fluorescent, and these fluorescent droplets are then selected and collected. You can even select for the highest producer cells to increase the quality of the cells you select. The positive hit droplets are then dispensed, one droplet going into one well on a plate which has been preloaded with cloning media or lysis buffer. And this results in a plate containing all of your potential drug candidates. And all in all, this takes around 6 hours, so this entire process to screen millions of cells happens within 1 working day. And how does this compare to the traditional method of limiting dilution? Well, instead of ending up with only half of your wells being occupied by cells, and even less of these being your cells of interest, with picodroplet technology, we're left with a plate whereby every single well has a cell of interest, not just a small proportion of your plate. So instead of having your population spread across tens of thousands of plates, you can collect your positive hits in a few high-quality plates. And this is a massive save on time, space, and reagent cost. And whereas limiting dilution requires a whole team of scientists working hands-on for long periods of time, our picodroplet technology only requires one user and is almost fully automated, so scientists can get on with their busy schedules. The decrease in manual handling also reduces user error and contamination issues. Picodroplet technology has also the added benefit of enabling new bioassays due to the unique microenvironment created by the picodroplets. These have opened up a gateway for high-throughput novel assays. And in action, the Cyto-Mine does exactly that. Here is a case study of the Cyto-Mine in action with Fujifilm Diosynth Biotechnologies. They found that the Cyto-Mine halved the time taken with their existing workflow. So project timelines were reduced by 3 months with picodroplet technology. And not only were the projects completed faster, the quality and quantity of molecular product were enhanced. So this enabled them to quickly identify valuable cell lines and successfully scale them up for biotherapeutics. And whilst today we focused on our applications in antibody discovery and a little bit of cell line development, our instruments are also applied in many other areas like cell engineering, cell therapy development, and many more. And if you'd like to find out more, please visit our website or get in contact. Our team would love to talk to you about the work that we do. So thank you for your attention, and I hope you enjoyed learning about the power of picodroplets and their role in single-cell analysis and screening.
Thanks so much for watching this Teach Me in 10, and I hope we'll see you again very soon.